Well, welcome. My name is Wade Webster. I am from North Dakota State University. I am the state extension soybean pathologist.
Good morning. I'm Damon Smith. I'm at the University of Wisconsin-Madison, and I'm the field crops extension pathologist here.
Hi, my name's Martin Chilvers, a field crops pathologist here at Michigan State University.
And today we're going to be talking about white mold. So to begin with white mold, we're just kind of walk over a brief overview, try to make this a little more conversational, talk about the biology a little bit, and walk through the disease cycle of this disease here. So initially, Sclerotinia sclerotiorum is this pathogen that causes white mold. It is a fungus, so it is very similar to many of our other crop diseases, but it is distinct in the fact that it can cause disease in many different plant species. Most, if not all, of our broadleafs. So in the northern Great Plains, we also see issues with sunflowers, pulses, and dry beans as well. It's very highly dependent on the environmental conditions. So this is both temperature and moisture. It needs relatively cooler temperatures, which we'll get into, and then some sustained moisture on that soil surface and within that crop canopy in order for that fungus to develop. And it also survives as what are called sclerotia, and these sclerotia live in the soil and they are able to surviving for up to 5 to 10 years in certain conditions. So these white mold sclerotia, this is kind of the starting point when we're thinking about the disease cycle. These are the overwintering structures. I typically like to talk about them as hibernating. They are not always going to wake up from year to year, but they will have sustained longevity within that particular soil. Now, these sclerotia are very important because they form the next structures, which we call apothecia, which we'll talk about here in a second. But it's important to note that the larger the sclerotia are, the healthier they are, the more of these apothecia are formed, leading to potentially bigger disease issues in that field.
So Wade, I got a question on that. Do you know like how many apothecia can actually come off a certain size sclerotium? Do we know that?
I don't know if we know the exact number based off of the size or mass of those sclerotia. It's definitely something that I'm wanting to look into. There has been work showing that different sizes produce more or less depending on the size of the sclerotia themselves. And I'm sure that there's a lot of environmental factors that play into that as well. And then
Yeah. Yeah, I think it's important to note too that I've seen some of these sclerotia give multiple flushes of apothecia too. So not only varying numbers, but they continue to sort of germinate over a period of time as well.
yeah, I mean, I think of these sclerotia kind of as like seeds. They store a bunch of energy in there, and if they don't use up all that energy in those apothecia, then they can still, like you said, have those second flushes. So for this infection, it's really important understanding when these apothecia form and they create ascospores that are released into that crop canopy. They need flower tissues to be present on these plants. And they need these flowers because the flowers contain a specific compound called pectin that the Sclerotinia uses to cause that initial infection. So what I call the susceptibility window is from about that R1 until about the beginning of that R4 growth stage. So when flower tissues are present, oftentimes when they're starting to fall off and dry up, um, is when that infection is going to occur. And most often around that node where those flowers are present and they're just starting to form or they get stuck on the end of those developing pods. Now, one of the really difficult things with white mold is that that infection occurs about 2 to 3 weeks before we start to see disease development. So what happens is once that fungus does infect, it starts to colonize and infect throughout that stem tissue of that white mold or of that soybean plant. Once it's in there, it does take that 2 weeks to burst out to show this white cottony growth on this outside here. Once it does form disease, then we start to see that bleaching of that stem tissue, which is the fungus killing off those plant cells within that stem. We also see the new sclerotia being developed here. So that's that next generation of sclerotia being produced. And in very severe cases, if you can imagine that entire plant is getting girdled off, water is not able to move upward within that plant. And so we see this kind of severe premature wilting and death of these particular plants.
Wade, this would be a good point just to think about that, or maybe emphasize the timing of fungicide applications. I don't know about you guys, but very often I get calls in August. Hey, I'm seeing some white mold. Can I get out there and spray? And very often when we go and look, they're at that R4 growth stage. And so people are pretty keen to sort of make some sort of revenge, fungicide application, but it's really not going to do much, right?
Yeah, we've actually looked at that in some replicated experiments, Marty, a few years back, where we actually came in at the R5 growth stage and applied a couple of different products, and then followed the disease over time, and then also looked at yield. And basically, once you get to R5, you just can't check it up, right? Like the damage is already done, and you're already too far behind the eight ball. So that's a good point. Like you can actually look at that early R4 growth stage and put some product on at that point, but that's a little bit like playing with fire. That's a pretty narrow window, right? So we really like to have folks focus on that R1 to R3 timing. I also had a question for you guys about just varieties and what you all see. In my location here in Wisconsin, we see a lot of indeterminate or semi-indeterminate varieties where you'll have pods forming and you still have flowers at the top node, right? And what's your feel? I feel like, at least in our observations, we still get a continuum of infections even later on in that season at those more upper nodes. And sometimes folks sort of translate that like white mold is just running down the rows through sort of contact, but I still think we're having some flower infections even late in the season. What's your take on that?
So generally, what we've seen is most of the infections happen down lower here in Michigan. Certainly not disputing that you can't get later infections. Coming back to that question of fungicide timing, I mean, if you want to try and control those couple of upper nodes, that's totally fine. But in terms of yield contribution or yield protection, you're talking about pretty minimal impact, right? Compared to that, Wade showed that picture of that stem down low that's taking up a whole plant. So,
Yeah, I agree. I don't think it's worth trying to focus on those, but it is also important for folks to realize that the longer that indeterminacy, the longer the risk window, but those are probably
mhm,
contributing, to yield not as much, right? And
mhm,
and so still focusing on that R1 to R3 timing, I think is best, and don't like try to convince yourself that you should be coming back
mhm,
in at R5.
absolutely. Have you
Yep.
seen anything different, Wade? So you're dealing with earlier maturity groups. It's quite different to what Damon and I, for the most
Yep.
part, are dealing with.
Yeah, I think one of the biggest drivers of those kind of late season infections is more canopy closure. We are fighting with specifically on the eastern side of the Dakotas over here. We are getting our beans in pretty late sometimes. We're not getting full canopy closure until the end of July, and
Mhm.
many of those beans are already out of that R3 growth stage window. So we definitely have seen some of those high hits. Now, if we're planting pretty narrow row spacings, then we see a lot more of those lower hits for sure. But it is kind of case by case, and it also kind of dependent. Talking about that canopy closure, this is really important for those apothecia to form. We need that full canopy closure along with the increased humidity and the moisture within there. So thinking about Damon, I know you brought up the varieties here, but I think it's also important to consider the kind of the architecture of that variety that you're planting as well. So if you are having a bushy type of bean, then that potentially could lead to greater white mold risk or susceptibility there comparatively to a more upright soybean variety. So after that infection occurs, we can imagine more infection. We're getting more sclerotia being developed, and these sclerotia here represent again the next generation. So again, thinking of say 2024 infection, these new sclerotia will potentially be issues for the 2025, 2026, and so on soybean crops here. Now these sclerotia, again, looking at this, you can see what it looks like on a plate growing and how big that these can get, but these sclerotia can also form on the inside of these stem tissues as well as the outside. So they can oftentimes get collected with the grain, get taken out of the field, but those sclerotia inside that stem or that fall off naturally from that are getting spread back out into that field, potentially serving as inoculum for that next soybean crop here. Now that we understand kind of the biology and the disease cycle of how this pathogen, this fungus, is able to infect into soybeans, how exactly is this causing yield loss? You guys have any comments here? Yield losses issues that you've seen?
Why
Uh,
is it why is it not linear, Wade? I mean, you would think like the more white mold, it would be a direct relationship, but like I've seen it not. And you hear farmers talk about, having a little bit of white mold actually tells them that they probably have a good high yielding crop.
Yeah, I think it's a part of what you guys are talking about with depending on when it gets hit, what time of year, what growth stage these infections are occurring. If we're seeing some on those upper canopies, that is that going to lead to severe yield losses or not? Um, Definitely variety selections as well. Like, are we seeing some of those some infections that are just not leading to that yield loss than what we'd see with other varieties? Um, there's a lot of factors here that play into it, but I think one of the bigger drivers is that soybeans are just such a plastic crop. They can adapt. They can kind of fill in the gaps where some of these dead plants are, and kind of make up some of those hit beans.
Yeah, I think some of it's also related to the fact that, our modern soybean varieties have a different architecture, right? So, we talked about that canopy closure, and a lot of that canopy closure, I think, comes from the number of lateral branches we have now on the modern soybean variety. So we have these bush type soybeans, and they're very different from what, like our grandfathers probably grew, right? Which were basically a straight stem, and all the pod yield was on that main stem. And I think some of the reason why we don't have this direct relationship is that sometimes those hits are on those lower lateral branches, right? And so the plant can sort of sustain that kind of damage, and it's not as big of a hit to yield as if you got one of those big long girdling, main stem hits. That's going to take the whole plant out, and that's a bigger hit on yield. So I think that probably has something to do also with this whole yield loss relationship, or lack of a direct relationship, I guess.
Yeah, I think that's completely right. Yeah. So within my extension talks, this is an image that I like to use. So this is a field that we had harvested a few years ago. And this is really important because there's a lot of different things going on here. Okay. So the first thing that we can see are these sclerotia that are present in this sample. So again, this is getting taken out with the grain. So these sclerotia, one, they're foreign material, so that could be potentially discounts at the elevator. We're also going to see some shrunken seeds. So these shrunken seeds are if those plants they prematurely died, they're not able to completely fill those beans. So those are going to be again reduced yield and 100 seed weight. And then we're also going to see some of these seeds, what we call mummies, which are infected seeds with that white mold fungus, covering those seeds that disease, that fungus had infected into those pods and started to degrade and prevent those beans from fully filling. So across the board, this one handful has multiple different points in which we're seeing yield loss here. Now, this is a figure here showing the relationship between the severity of disease. And Damon, this kind of goes into your comment of why do we not see this linear relationship between disease severity index, which is a factor of both the percentage of plants infected as well as the severity of the infections, as a relationship to yield. And so you can see this clear relationship in which we get to about 0 to maybe 40% range, in which we're seeing pretty minimal yield loss in this red and green line, which are the estimates. Once we get to about 40 to 60, 65 or so, we're seeing some more yield loss. But after that 65% range, we're seeing some pretty substantial yield losses. And across this particular analysis, we're seeing about every increase of 10% of disease severity index after about 65%, we are seeing about 10 bushels lost to that. Any thoughts here, Damon? Kind of going off of the yield losses.
Yeah, I think that just comes back. There's a lot of spread here in the data, and we often get asked about, why is that not tighter around those estimations? And that I think that all comes back to varieties, right, and locations, and we have multiple maturity groups and things in here, but you can see the relationship. Yes, you got a pretty wide band around that estimate, but those relationships do seem to hold true all the way through, right? And I think a lot of this again comes from, lateral branch hits at those lower disease index scores, right? That playing a role in there. Certainly, we're probably having some of that smaller seed, like you showed, and chalkier seed, maybe at the lower disease index scores, which are not as substantial as if you take out an entire plant and that plant actually can't fill out all the seed, or we have complete seed loss on a particular plant. That's what happens with those higher disease index scores. So I think that's probably what explaining some of that. And again, the breeders have sort of changed the architecture of these plants, and a lot of it has to do, I think, with those lateral branches.
Yeah, that's great. All right, so now that we understand the disease itself and the kind of yield loss that associated with this, we're going to jump into some cultural management.
Yeah, so let's chat about variety resistance, row spacing, plant population, fertility, and some of the multi-state work that we've done. Go to the next slide, Wade. So variety selection is really important, like we were just discussing, and this is just a nice photo out in the field demonstrating, a susceptible variety there on the right-hand side compared to something that's got better resistance package, standing up to that white mold a little bit better. Next slide, Wade. So this might be a little bit of an oversimplification of the data we just talked about. It's not necessarily linear. This is, in a more narrow maturity group soybean set. So we've got about 10 or so different varieties here screened over a couple of different seasons for a company. I just like to put this up because it demonstrates a couple of different things to growers. It demonstrates the difference between seasons. And so what we're looking at here is the white mold disease severity along that bottom axis and then yield. And there's a correlation like we just talked about. It might be a little more complex than this. But within a year, you'll see a spread from most resistant, least amount of white mold development, greatest yield through to those developing the most amount of disease and losing yield because of that. And so my point to this slide is that within these couple of years, you could be looking at, 10, 15, 25 bushel difference between a variety that's really susceptible compared to something that's got a better resistance package. So variety selection is really important. And I know, we're doing a lot of work as well, looking at new varieties. Damon, you guys are trying to do some breeding work as well. We're doing breeding here with our soybean breeder to try and find better sources of resistance to white mold. And that's a real struggle there as well, and a real key to trying to move things forward.
So Marty, just talking about resistance and the types of resistance, what do you think's going on, especially in the field when you look at data like this? Do you have a handle for sort of how that resistance is manifesting?
Right. So you're getting, yeah. So resistance is complex and we're sort of dancing around. So what we're talking about here is what we might call physiological resistance, where we get an infection, but the infection is limited to one spot on the plant, versus where you get, movement through the plant and kill, death of that plant. And then there's avoidance through canopy architecture. And then there's yield potential as well, right? So it gets a little complicated because there's differences in yield potential between these different varieties. So it's not super simple, but there's complexity to it, right? And it's challenging to breed for as well, because it's not just a single gene that we can take and confer resistance to white mold. It's multiple gene controlled, and so that's very challenging in a breeding sense.
Do you think there's any factor of the white mold pathogen itself? Do you think there's like different populations out there impacting the?
Sure.
Yeah.
Yeah. I mean, yeah, some of that multi-state work, I think was that Jamie or one of the other grad students looked at making sure that were screening with multiple isolates of sclerotinia, not just taking one isolate and using that to screen everything, because that's not representative of the diversity that's out there and the ability of that pathogen to overcome, potential different resistance mechanisms.
Yeah, that panel I think was really important in giving us some confidence that the resistance we were seeing was actually real. Because sometimes it's hard to understand whether if you screen with one isolate whether that resistance is going to hold up if it goes to a different environment, right? Because we do know that the isolates do vary. We're actually taking a look at the populations right now using whole genome sequencing, and we actually have some isolates from you all in that collection, and we see a great amount of just whole genome genetic sequence differences, right? We don't know what that actually means yet. We're trying to dig into what that really says about how aggressive, for example, these isolates might be, but we can see genetic differences, and they do seem to correlate mostly to actual location. So I think there's probably some adaptation of some of these isolates to certain locales, right? And that might drive, how they interact with different hosts, which makes the whole resistance thing even more complicated.
Next slide, Wade. Thanks, mate. So this was some work looking at the effects of row spacing. So looking essentially at 30-inch versus 15 or 14-inch row spacing and how that might drive white mold development. So here we're looking at the number of apothecia in those red lines there. And essentially what we were seeing is in that more narrow row setting, we're seeing apothecia developing a little bit earlier, and then a greater number of them. So in this particular year of study, we saw about 60 times more apothecia in the 15-inch row versus that 30-inch row spacing. So row spacing is an important component. We just talked about the microclimate and canopy closure being very important. So row spacing is one of those. Next slide.
So Marty, just a comment about that. And this year was a really good year. The 2024 season was a good year to see this in Wisconsin, at least. And that, if you have high populations of soybean cyst nematode, which delays that canopy closure, especially on a wider row spacing, it really deters white mold development, right? So we got a good look at that because we had a lot of sort of delayed row closure. I don't know about you guys, but in those situations where we could make it into, even into, mid to late R4 before the canopy close, that really sort of shut down the whole white mold situation too, right? So that sort of tells us how important that canopy closure really is with the whole apothecial development thing, right?
Yeah, absolutely. It's really key. And Wade, I think this is some of your PhD work, right? Where we're working across states to generate data. So expanding upon that, looking at that 15 versus 30-inch seeding rate and the interaction. So Wade, do you want to go ahead to the next one? So looking at different seeding rates here, looking at from 110 up to 200,000 seeds per acre, how does that impact white mold? And really not a great deal of surprise here. Those higher plant populations, we start to close canopy a little bit earlier. And you could also think about down the row closure versus, between the row, there's a little bit of complexity there as well. But the higher plant populations or higher seeding rates led to greater white mold development. I just grabbed a photo that Mike Staten had, one of our retired extension educators, just showing on farm here, 80,000 up to 160,000 seeding rates, and just those differences in the amount of white mold that you could see from the drone imagery.
What were the yields in those, Marty? Oh,
Glad you asked, Damon. Lovely. So Mike ran some economics on this as well. So this would have been at the $13 per bushel price. We haven't seen that recently. But yeah, essentially under these heavy, and we've got to stress here, I'm a little nervous to promote 80,000 seeds per acre as a seeding rate. I'm not an agronomist. I'm not going to, get into that whole deal. But basically at these lower plant populations, though, we out yielded those higher plant populations. So certainly if you're out there planting 160,000 seeds per acre, I would really give that some thought and try to drop back to 130, if not 100. Again, chat with your agronomist about how low you can go. I'd be a bit scared about 80 if we don't have, we get phytophthora or something else going on. Now all of a sudden you're in a replant situation. But under heavy white mold, it can definitely pay to really pay attention to those seeding rates.
Yeah, we've been having that conversation with our soybean agronomist here, Sean Conley, and he seems okay down to 100,000, right?
Yep.
I think the caution that we all worry about, and you mentioned it, is if you got Phytophthora or you're worried about Pythium, these kind of early season seed or even mid-season stem rot problems. You really do need to probably invest in the seed treatment. Make sure you don't skimp in that fungicide seed treatment on those beans, because you really do need to get every bean out of the ground when you're at those lower populations. But we did some work just looking at fungicides with and without on these high and low seeding rates. So that you could almost negate the need of a fungicide in the lower seeding rates. It's really quite significant. Yeah,
absolutely. Yeah, so I mean, this is just demonstrating that yet again. So we've got on the left there that 15-inch row spacing. There's more disease rate. We're able to suppress that with a fungicide, and on the right hand side that 30-inch, so a little bit less disease there, and again with a fungicide, knock that down even further. Next slide. All right, and then integrating these as well. So putting together the row spacing. And looking at final yield. So here's your yield data. Surprisingly, I guess this is a little counter, right? So I guess, I don't know what you guys want to say about this in particular, but there was an impact here, I guess, of plant population, at least in that 15-inch row spacing on yield. So being mindful of that, a little bit less so in that 30-inch row spacing. But some impact there as well.
Yeah, I mean, it's a little hard to tease out in these smaller plots, and we've had that conversation about just what does this mean, and I think what it means is like if you look at an acre of soybean production, if you plan for say 140,000, maybe on the 15-inch row spacing, if you have precision plant technology, move that 140,000 around on that acre, right? Like, don't plant the same populations in your high white mold areas versus your low white mold areas. And a lot of us know where those are. So, maybe thinking about this a little more on the precision plant side of things and how you move that around. Again, it's just sort of hard to see because this was a lot of small plot work across those locations. But as we started to look at longer strips and things, you can start to see the advantages of the lower seeding rates, even on these narrow row spacings. And I get this question a lot. I don't know about you guys, but we have a lot of water hemp here in Wisconsin. And I take a lot of baloney when I start talking about maybe wide row spacings and low plant populations and all that sort of thing. And I'd love to hear your opinions on this, but we've been talking with our weed scientist Rodrigo Worley here, and we sort of come to the conclusion that we can balance water hemp and white mold in the same field by maybe going on the 15-inch row spacings, but dropping those planting populations, right? So maybe a 15-inch and looking at 100 to 110,000 and your planting population may be a good balance there. I don't know what your guys' opinion is on that.
I agree. We have mare's tail and other weed issues as well. So I don't think you want to give up on the 15-inch row spacing necessarily. Right. Like you said, pull back on populations and consider that in the mix. Yeah, I think that's really critical. And the other thing, I guess, in here, we haven't incorporated the economics piece, right? And right. This is a hot topic at the moment with lower soybean prices, and so we really want to factor that in the seed cost, and that's a really important determinant as well.
Yeah, I mean that's a good point, Marty. You can have all the yield in the world, but did it cost you a lot to get there, right? And
Hmm.
sometimes even with slightly reduced yields, if you had a lower seeding rate there, the economic return actually is higher in those situations than it might be where you're just, pushing yield to the nth degree, right? So, yeah, making sure you really turn the numbers and be honest with yourself.
That absolutely. Yep. All right, next slide, Wade. And fertility was something else that we had a look at. So again, just back to canopy closure. I've been involved in some discussions over a few years now with a really good grower. He does an excellent job on corn, but he takes that same mentality to soybean. So he's pumping in starter, fertilizer, and then sure enough, he's got white mold issues, right? So we're like, dude, like let's pull back, let's cut back on the starter. Let's not treat it the same way we treat corn. And through that, and actually treating some of those areas that we know we're going to have white mold with a fungicide, and being strategic, we've really come a long way in his management program. And this is just some, multi-state data to demonstrate that, yeah, high nitrogen levels, high fertility levels, drive canopy closure like we've been talking about. And because of that, drive the white mold.
Yeah, Marty, I think one of the things that I see, at least up here on just a little bit east of us, we got a lot of dairy farmers. They're spreading that manure out there. And, there could be a lot of interactions here, but I think that manure for the corn crop is kind of driving some of this canopy closures you mentioned here with this fertility.
Yeah, absolutely. Yeah. Next slide. Thanks, mate. So this one's kind of interesting. So the tillage component. So there's been a recent study here just published, maybe that was 2023, but anyway, um, it's just demonstrating, and I've got that figure up there, demonstrating greater sclerotia survival on the soil surface as opposed to being buried 3 centimeters down, or an inch and a quarter or thereabouts, inch and an eighth. So after 2 years, I was still able to find about 15% of those sclerotia being viable on the soil surface, as opposed to being, just lightly incorporated. So this sort of gets into the whole conversation about tillage and what's best. There's other studies out there showing lower apothecia counts in no-till, and sort of metadata type studies showing that white mold is less prevalent in no-till. Something that we've sort of recommended here in Michigan, and actually we've applied it to some of our own research ground where we don't want to have white mold, but we're pushing irrigation for other diseases, is to come in and no-till something like a wheat crop into those soybeans to try and keep sclerotia on the surface, or at least close to the surface, and then have that non-host crop like that wheat crop or a small grains crop to promote sclerotia germination, apothecia production, and trying to burn out those sclerotia. So it's just kind of interesting to see that, it's not, the story's not necessarily quite that clean. Maybe perhaps putting, some light tillage and incorporation of those sclerotia could be helpful, but we have to be very, very careful. Moldboard plowing, we don't tend to want to recommend that from a soil health perspective and erosion and whatnot. If you're going soybean on soybean, maybe that's an option to try and bury some of those sclerotia down for that next season. But we don't want to see you guys doing soybean on soybean anyway for white mold and a whole bunch of other reasons. I don't know what comments you guys want to make on this tillage side of things, but it's definitely interesting. There's a lot of complexity to it.
I think we need to go back and kind of look at this, Marty. And I think one of the things we need to account for is actually soil type and organic matter.
Mm-hmm.
Because I think, in my opinion, at least, the observations that we've made, and we actually have a couple of experiments now deployed to try to address this. We have widely varying soil types here in Wisconsin, from basically beach sand with no organic matter all the way on up to, some pretty high organic matter, deep prairie soils. And what we've sort of observed is, on those deep prairie soils, there's a lot more competition, microbial activity, and I think that affects survival of these sclerotia, right? And so that could be driving some of what what we see in terms of differences in till versus no till and what to do. I think soil type probably is driving some of those, and we're trying to get at that to see, does tilling on, say, beach sand, affect survival versus tilling, on a nice big deep prairie soil. So we'll see how that works out. But I think going back and kind of looking at soil type might help us wrap our head around some of that.
Yeah,
Yeah.
Damon, I've at least observationally have seen very similar things up here as well. Those heavy sand soils are much more white mold infested than some of those higher organic fields are.
Yeah, interesting. Yeah, so that brings us to chemical control, and we've sort of been working it in here a little bit through the discussion, but we'll kind of dig in here a little bit. And of course, just a shout out to both my colleagues on this call as well as Darren Mueller and Darcy Talanko. They all made contributions to the status set as well. So Wade, next slide. So we've had a series of uniform fungicide trials now for over a few years, and I'm just going to show you a subset of these trials and kind of focus on this 2022 field season because there's some interesting things in here where we looked at the same treatments both in Hancock, Wisconsin, and then also in Michigan with Marty's group here. And ran these trials together. And what's nice about these uniform trials is we can establish or get a lot of data very quickly across a number of site years by running these uniforms. So again, a shout out to our colleagues. Wade, next slide. So here are the treatments and also the severity index scores here. So again, we have two locations that make up the means and then the variance that you see in that little error bar at the top. And some interesting stuff going on. The non-treated check is in that yellow box, kind of to the upper end of the disease severity index scores there. And then everything inside the green box to the left-hand side of your screen, those were among the best treatments. And from a statistical standpoint, really not different from each other or different from the best treatment out there. And there's a couple of takeaways here. First is that timing is very important, right? So we talked about that earlier on that we have to have an application going on somewhere in that R1 to R3 growth stage is really the optimal. We can use forecasting tools. So we have our Sporecaster app out there, which can help with that timing. And that can actually help optimize within that R1 to R3 time frame where to apply that product to really get maximal benefits. And you can see in that third bar over from the left, we have a treatment in there, which is Endure at 8 ounces at R1 followed by R3. I by no means would recommend this as an actual treatment, but what we do with that treatment is we put that in there sort of to say, okay, here's the best case scenario, right? We're covering all the way from R1 to R3 in terms of fungicide protection. And can we find a treatment that actually matches that? So it serves as a positive control, if you will. And you can see Endura at R3, or at least using Sporecaster, which turned out to be mostly R3 in both these locations that year, or even a 2-pass in some cases, was pretty good. But also we have to account for how we're actually applying the product, right? So we've got a couple of interesting treatments in there with 360 undercover applications. So one of the downsides of actually coming in sort of later in that bloom time near that R3 is now we're dealing with a very dense canopy, right? And so trying to drive fungicide down in the canopy really becomes an important thing to consider. And if you look at a product like Omega, it's a very good product, but it's a contact-only fungicide, right? So it has very little mobility in the soybean plant. And so coverage is everything. And you can see we can make Omega look very good if we sort of account for the application, right? So if we put it on with a 360 undercover, you can see it really improves the efficacy of that product and drops the disease severity index scores. So thinking about all of these things, if you're going to go late in that bloom time, really think about, your application strategy. And there's other ways to sort of drive fungicide in the canopy. You don't necessarily need to put it on using some sort of drop nozzle. setup, but maybe increasing pressure or increasing the water volumes that you're actually going to apply at those later growth stages might help improve the efficacy of those products. I don't know if you guys have anything to add here.
I mean, yeah, I agree with your comments, Damon. I guess you and I see maybe a little bit of difference in our trials, and maybe that's because of some of the row spacing, right? So we're on 15-inch row spacing. So sometimes we see better efficacy from that R1 than the R3, but not always. So it's also, again, back to Sporecaster, based on when those apothecia are actually coming up and, producing spores for plant infection.
Yeah.
Um,
I mean,
Yeah.
I think to your point there, Marty, it's a lot of that canopy closure. If you're getting there, like you said, damage, we need to get penetration of those products into that canopy. And so, Marty, if you're waiting until that R3, that canopy closure might be so dense that we're just not getting that product in to protect those flower tissue, right?
that's right.
Yeah. Next slide, Wade. So here's the corresponding yield data from those same trials. And first off, you'll see the better treatments tend to give us more yield return. We would expect that. And so everything inside the green box, those are all statistically similar. So what that means is if we were to repeat these experiments, anything inside that green box, they might reshuffle with each other, but they would tend to be some of the better treatments in those trials. And again, where we're taking some time to really consider the timing and the application strategy, we're getting good return on yield there with those treatments. We haven't really talked about it much yet, but there's also other products out there besides fungicides. There's actually an herbicide called Cobra out there that does have suppression of white mold on the label, and we've all played around with that product a little bit. It seems to be, at least in my trials, it seems to be a decent product, especially in the heavy white mold areas. I would caution the use of that product maybe in what you might consider a light white mold or where you're just seeing sort of spotty areas, because you can lose a little bit of yield where you use 6 ounces of Cobra, say at that R1 growth stage. So that is something to consider. It does seem to control white mold quite well, but you can get a little bit of yield reduction that you might see, especially if white mold's light. If white mold's just really heavy, though, we tend not to see any yield reductions out of Cobra, and in fact, they tend to be pretty good looking treatments in some of those trials. I don't know, Marty, if you've seen that in your trials as well.
Absolutely. Cobra tends to be as good as our best fungicide product under
Yeah.
those heavy white mold years. I totally agree, though. You've got to just put that bit of caution in there under light white mold situations. Yeah, you may be dinging yield. And actually, what was really interesting and nice with this multi-state work is it's hard, with a single site or a single, couple of years to actually pull out that ding from Cobra. But when we did this multi-state, we we're actually able to demonstrate and put some statistics to it. So that was really helpful just to put that cautionary note and, yeah, help people understand that, I guess.
Yeah.
I
The other,
guess one caveat to Cobra as well is that we can't apply it the exact same like what we would our fungicides. You can see here these are at V5, right? So making sure that we're not dinging those flowers up like we
Yeah,
would some
you
of our
can.
fungicides.
Yeah, you can go a little earlier, although you can't go super early. We've played around with that as well, right? So, that there's a couple of things going on with Cobra here in that you're modifying the canopy to a certain extent, but I think actually some of this work was done at Michigan State, I think, Marty, a few years ago. I think
Yep.
Ray Hammerschmidt, I think, did some of
Yep.
that, looking at lactofen, which is the active ingredient. And not only is there canopy modification, but there's an upregulation of induced systemic resistance in these plants. The problem is that systemic resistance sort of ramps up and then it can go back down, right? So if you go too early, you sort of miss the benefits, right? And we've done some of those treatments looking at, say a V2 application, a product to try to upregulate that. But it seems to be by the time we get to kind of that bloom time where we really need the protection, it's already burned off, right? And doesn't give us any advantages. So Wade's point about making sure we're careful about that timing of that product too is really important. Yeah, next slide, Wade. Yeah, and so just to look at some other data here, and the gray bars are kind of covering up some of the disease index data here, but this shows you that as you have higher disease index, you get less yield. It moves around a little bit, but some of the better treatments are actually in the green there. Looking at Endura again, that seems to be sort of our benchmark product out there, and then applying that product using a good timing tool like Sporecaster can help out. Sometimes a second application might be needed. We've been looking at that, and we've been looking at things like Mirvis Neo for the follow-up to give us some, say, frog eye leaf spot control, and that seemed to work out in the 2024 data set as well. And then one other thing we've been starting to play around with here in Wisconsin, I know you guys probably are looking at this too, is we're revisiting some of the biocontrol products that sort of have some efficacy against the sclerotia, right? So at plant, applying, say, contans, which can help reduce the sclerotia. We didn't necessarily see many benefits out of that application in this 2024 season, but we're going to continue to work on that and try to understand, again, how does soil type impact that application or product, and some of the other factors there. But certainly a possibility in terms of actually trying to go after the inoculum for this fungus rather than trying to protect the plants early or later in the season. So just a different strategy to sort of think about there. So we talked a little bit about just application technology and the real need for getting good canopy penetration. And we've gotten a lot of questions here in Wisconsin about drone applied fungicides in soybeans, mainly in white mold areas where the field's really kind of boxed in, wooded areas, things like that, where it's hard to get a big sprayer in. Taking a look at these drone applications, and so we've started to do this. This is a 2023 data set where we did some larger strip trials, looking at our 2 gallon per acre drone applications at our R3 timing using Endura at 8 ounces, and then comparing that to a conventional 15 gallon per acre ground application rig. So Wade, next slide. So this just shows you what that looks like. We've got the drone flying side by side with the ground rig there, and you can see the ground rig, applying a 90-foot swath. All in one pass. One of the downsides of the drone is that you're really looking at a smaller pass. So it did take 3 passes to get the same width as it does to get 1 pass of the ground rig. But the other thing you can see is you really can't tell where the drone's actually spraying, right? You can't actually see the spray, whereas with the ground rig you can. And so one of the things we were interested in seeing is, can we match, even at 2 gallons, can we at least match the efficacy of the product that you might get out of applying at 15 gallons per acre with the ground rig? So next slide. And so these are just some aerial images on the left-hand side. You can just see the general natural light damage from white mold. You can see the different replicates of these large strips. And then on the right-hand side, we've got NDVI. So where we had, of course, more white mold damage, you're seeing more damage from, or more open canopy and presumably more damage from the white mold fungus there. Next slide. Here's the actual disease incidence numbers. So drone and ground are in those lower bars there, and you can see definitely some reduction where we put some fungicide relative to the non-treated. But what was surprising, I guess, is that we were able to actually match the ground rig in terms of disease index or disease incidence reduction, right? So kind of surprising. Remember, we're putting 15 gallons per acre out, which would give us good distribution of that fungicide product on the plants, versus 2 gallons per acre out of the drone. And so what we're thinking is going on is actually the downdraft from the drone, and the 4 propellers that it has on it are actually helping to push the fungicide down in through even these denser canopies at that R3 timing. So next slide. And those are the corresponding yields there. You can see, again, where we had the fungicide applications, we were able to protect yield And in fact, the ground and drone were actually comparable in terms of yield there. Now, am I saying that we should just go away from ground rigs and go to drones? No, I don't think that's the case. But I think what this tells us is that indeed in some of these tighter fields, or if you want to again do some precision control of white mold, so in other words, mapping where white mold is in a particular field and only treating those areas, the drone is what we believe to be a comparable option to actually going out and maybe going across an entire field with a ground application rig. And I think this also might be a sort of a middle ground in terms of, we played a lot with drop nozzle technology, and we showed you some of the 360 undercover technology there. That seemed to help, of course, with our efficacy, especially for our contact products. But dragging a nozzle that's on a drop through R3 soybeans causes quite a bit of damage. And so the drone might be a good option there if you're wanting to ensure good canopy penetration, lower carrier volumes. The drone might be the good middle ground there. So the other thing I just wanted to mention too is that there's lots of places to find fungicide efficacy data. Looking at trials like myself or Marty or Wade, but we also get together and through the Crop Protection Network, we compare our efficacy ratings each year, and we combine those every year into these fungicide efficacy charts. And a QR code there will actually take you to the CPN fungicide efficacy chart. And in fact, on that chart, we include white mold, so you can actually find the products that have the best efficacy ratings there. So that would be definitely something that you'd want to check out if you're trying to consider a particular product for use. That's a conglomeration of all the data across all the different states.
Yeah, and Damon, just to jump in there, I can't emphasize enough when you're dealing with white mold to go and look at this chart, because there are some products that are labeled for soybean and they basically do nothing for white mold. It's kind of the counter when we're thinking about maybe some other foliar diseases. Maybe most products do a reasonable job, but for white mold, it really does matter. So please pay attention to these charts.
Yeah, good points there, Marty. Next slide, Wade. The other conversation that's coming up this season is just return on investment. Margins are really tight, and really trying to understand what are the break evens and what are my estimated return on investments if I make a particular fungicide application. And we actually have a tool that can help you with this. It's called Spore Buster. It's a smartphone application. That QR code will take you to this tool as well. And what's nice about this is this has many of the common products that are promoted for white mold control. We've taken some of the efficacy data and worked these into some models, as well as some of the economic return on investment models in here as well. And you can actually go in and put some inputs of your own in, so you can adjust that expected price of sale. You can adjust your expected yield on your farm, and then you can also adjust those treatment costs. So we put in some median treatment costs that sort of are default in the app, but you can get your local pricing and actually put those in this tool as well. And that'll actually recalculate using the background models that run this tool and give you some estimated break-even probabilities and then average net gains. And one thing I'll say about this is make sure you're looking at the break-even probability percentage first before you move to the average net gain. Because those two numbers sort of tell you different things. The break-even probability will tell you, what are the odds of actually getting your money back on that investment. And then if you do reach that break even, then what is the average net gain after that? So that's how those two should be used together. But this is certainly something that can help in an economic climate like we're in right now, trying to really understand how much benefit you might get out of some of these products. And the best product out there might be really expensive. And so you actually might see higher return on investment out of some of these other products, right? So that is something to consider on your farm.
Well, thanks, Damon. So we're going to kind of shift over to two more kind of newer perspectives on white mold management. This first one, and we've kind of talked about the use of genetic resistance. So resistant varieties or susceptible varieties, and kind of the differencing there between whether that is disease development or yield. But one of the things that is not really often discussed is the amount of sclerotia that is produced depending on a susceptible versus a resistant variety. And so these are some trials that we had run across Wisconsin and North Dakota between 22 and 2023, looking at two different soybean varieties. These are breeding lines, they're not commercially available, but they are genetic siblings. And one of the key differences between these sibling lines is that the 9144 is highly susceptible, while the 91-145 is highly resistant here. Okay, this is why we kind of use these two lines as a proxy for susceptible and resistant. Now, one of the things that we're looking at is the top portion of this table here is looking at disease severity index. And you can see that, again, the susceptibles are consistently higher levels of white mold comparatively to the resistant. But the most interesting portion of this story here is on the bottom side of this table, looking at that sclerotial production. And this is pounds per acre. And this is incredibly difficult to conceptualize as the density of these sclerotia is very, very low comparatively. Okay. But looking at say that Oaks site in 2023, that susceptible variety produced on average about 124 pounds per acre of new sclerotia. Okay. That is a lot of new sclerotia that could potentially lead to greater levels of infection, whether it's that next season. Now, same exact location for that resistant variety, instead of 124 pounds, produce a little under 6 pounds of sclerotia. Okay. So, we talked about this as white mold oftentimes shows up in little pockets within that field. It is not often widespread across the field. It can be, but if you're thinking about highly, highly concentrated amount of high levels of new sclerotia being put back into that field, okay, that's just increasing the risk, that probability of disease developing at high levels that next season. Now, to kind of take that a little bit or one step further, we actually put together some linear regressions from this data set, trying to predict outward how much sclerotia are going to be produced depending on planting a susceptible versus a resistant variety. And so with a susceptible variety, again, that risk of high disease incidence is going to be higher, but we got about 25, a little over 25 pounds per acre for every 10% increase in incidence. And so we talked about earlier that we're going to see, and this between incidence and disease severity index are slightly different, but we talked about that disease severity index having yield loss only after about that 40% mark. So if you can, again, just kind of translate those two, we're looking at about 100 pounds of new sclerotia once we start to see yield loss. Now, thinking about it from a resistant side, there's going to be fewer sclerotia, about 14.4 pounds of new sclerotia for every 10% increase in disease severity or disease index incidence here. And so this is just another portion of the story that is oftentimes not discussed as the amount of new sclerotia put back into that field. Any questions there? Comments on the sclerotia production stuff, guys?
It's an important long-term strategy, right? To try and reduce that sclerotial load.
Yep.
Yeah, and I think, as we think about return on investment, this is certainly something to consider, and it's hard to sort of put some numbers to that thing, right? Like, what does that actually mean in terms of subsequent disease? But I think, if you have that much reduction, we're talking about 10 plus pounds difference, from a susceptible to resistant, that's substantial. I don't care whether you can run statistics on that or not. That's quite a few sclerotia, right? So any way that you can really start to rethink this, and I like, I like thinking about this again, like weed scientists do, right? So this thing makes sclerotia, which are sort of like weed seeds. And if we can impact that sort of seed bank, if you will, this is a really great way of sort of, managing this problem long term.
And one of the other things here is that I guess the selection of your variety costs you absolutely nothing. We're not talking about the application of a fungicide. So you're reducing that long term for the exact same cost as you may for a susceptible variety here. So the next thing, and I think this is the last topic that we're going to talk about today, is going to be the use of biopesticides. And so I'm going to borrow a few slides from our colleague Darcy Telenko talking about biologicals. And this is an area that has been highly pushed over the past few years, looking at many, many new products for the application of biologicals or different microbial organisms to reduce white mold development. And we talked a little bit earlier, Damon did, on contans. We'll talk about that again just slightly here. So these biologicals generally are considered less toxic. We're not putting in that selection pressure against these some of our conventional fungicides. And oftentimes we need relatively small quantities of these. These microbes, they're capable of reproducing themselves and increasing in numbers if we give them enough the correct environment and conditions. But one of the caveat here is that these have relatively shorter REI comparatively to some of our commercial products, which may be of interest to certain individuals or operations. Now, this contans, okay, this is another fungal organism called Conotherium minitans, just threw the name together, which is contans, has been around for many decades, and it is very effective at colonizing these sclerotia. And so this is looking at it on peanut for sclerotinia blight, but the concept is the same. This fungus needs these sclerotia as a food source to eat and reproduce and survive. And so this is data that Darcy Telenko had completed years ago looking at this here. One of the points I want to look at is this 2002 data. The control is when these sclerotia had 0 contans applied and then 1, 2, or 3 years of applications of this contans product, looking at the development of disease, or what we call area under the disease progress curve, AUDPC. So the lower the bar, that means the less disease developed in that particular case. And so when we give these sclerotia exposure time to these contans product, they are able to be degraded and reduce that development of white mold development. You can see these here are the cells of these sclerotia on the left-hand side. The healthy ones, they are very much intact, but the ones on the right-hand side that have been parasitized by this contans fungus have been degraded, eaten away, and collapsed, making those non-viable. No apothecia are being able to be formed from these. Now, this is looking at that same fungus here on a plate, but again, these have been around since 1940s. There's a lot of information out there, but this is generally a product that is a little higher of a price point, and it needs to be applied under certain considerations. It needs to be applied and incorporated, and it oftentimes need to be stored under cool conditions as it is a live organism relative comparatively to our commercial, conventional fungicide products. And with that, I want to thank everybody for all of the work that they've been able to do to help put this kind of slide deck together. We have a lot of great colleagues across the Midwest and across the U.S. And thank all of our funding agencies and all of the checkoff organizations that have been able to support this sort of research so that we can get this presentation together. And lastly, I want to thank the Crop Protection Network for allowing us to give an opportunity to present all of this and to be a great resource for all these farmers for different diseases and pests that we deal with across the board.
And on the Crop Protection Network, we've got a web book as well on sclerotinia. So there's a lot of resources there for white mold management to take a look at.
Yeah, and don't forget to check out those fungicide efficacy charts there either. So lots of resources at the CPN Crop Protection Network.
Great. Well, thank you all for joining us today, and have a good rest of your day.